It is my pleasure to introduce you Professor Torsten Haferlach, head of Munich Leukemia Laboratory, also known as MLL, one of Europe's expert laboratories in providing molecular pathology assessment of hematological disorders. Professor Haferlach will tell us about streamlined next-generation sequencing workflow for targeted resequencing that they have established at MLL over the last couple of years. Professor Haferlach, please feel free to start whenever you're ready.
Well, thank you very much, Svetlana. Good afternoon, everyone, and thank you for joining this webinar. I'm happy to share with you our experience over the last years using next-generation sequencing in a routine setting in our laboratory for leukemias and lymphomas, and I will be also happy to answer your questions after the talk or by email. That will be also possible. So I'd like to start just showing up this slide that clearly gives the background that I would like to share my experiences, the experiences from the laboratory, and that does not mean that all the things I'm going to tell you are the opinions of Illumina. So it's the talk that we prepared here in the lab, and I will share with you what we think about and how we apply it. So as many of you may know, the methods that we use today to diagnose, to classify, and to follow up in leukemias and lymphomas are depending on several different aspects, starting from cytomorphology, which is a very old method at the beginning, including histology, cytogenetics, as well as FISH, immunophenotyping or immune histology, and more, and many of you will use that instead of the others, molecular techniques that came up over the last years being more and more important, including gene expression profiling and especially now the increasing field of next-generation sequencing. So what we do here in the MLL with respect to the different technologies, we use qualitative PCRs as well as nested PCRs, a lot of quantitative PCRs for follow-up samples, melting curve analysis, fragment analysis, and Sanger sequencing. That is still the backbone of our sequencing part of the laboratory. But more and more increasing and having a very quick way from research use only to the application in a routine workflow to diagnose leukemias and lymphomas, we now use next-generation sequencing from the Amplicon-based approach by the Illumina technique, as I will depict later to you. We used many years the 454 technology from Roche. We use RainDance and the Thunderstorm instrument as well as Fluidigm, and I will get into these different techniques in detail during my presentation. So you are all aware that this is a standard approach to go for molecular genetics, and this is the most important approach we do for the diagnosis of CML, for example, and especially for follow-up, because this investigation completely defines the treatment efforts, the treatment follow-up, and will change the drugs that are applied to the respective patients. So this is not an assay that we think will be in a very short time be put onto a next-generation sequencing system, but it may happen as well. The next question is, could we use minimal residual disease as it is needed with a very low sensitivity of 0.001 or even lower for BCR-ABL, also with next-generation sequencing today or in the next future? And I also will address this during the later slides. We all discuss this not only here, but we discuss this within the European Leukemia Network, where many centers that diagnose and treat patients all over Europe are connected, running trials and discussing about the data and the next ways to treat and to diagnose patients over the last 15 years. And within this European Leukemia Network, there is a working package called Next Generation Sequencing, as shown here, and I am the lead participant for this more than 10 years now. So this was called gene expression profiling in the first years when we started in 2000, but during the times the gene expression profiling stepped back in the interest for research and was not introduced to a routine method for many aspects, but in the last 5 years, next generation sequencing took over, and our interest is now much more focused on this technique within the work package 13 of the European Leukemia Network and also in our lab as well. Another thing is, and this is a very important aspect that is not clearly addressable for next generation sequencing today, but is one of the unmet needs for the future, how to standardize the molecular approach using next generation sequencing. And there is also very recently available since 2015 an approach to standardize BCR-ABL for minimal residual disease and use very specific standardized consumables to organize the different labs, to run ring trials, and to be sure that everyone more or less is measuring the same. So this is one example for standardization in molecular genetics, and so far we are far away to standardize all the different assays and genes that we interrogate and approach using next generation sequencing platforms, and this will be one of the future goals and future aspects that need to be solved. So I'm not quite aware if all of you are in that field, but I just want to give you the example of the AML in the last 25 years and what happened there. So what happened was that more and more genes were found to be of importance for AML, not only in the diagnostic setting, but also for routine and for prognosis, and more also coming up as targets for specific treatments. And this was between 1990 and 2005, and this is what happened due to all these next-generation sequencing and high-throughput sequencing studies that much more information came up in the last 5 to 10 years. That leads, and this is a figure taken from the TCGH paper from Tim Lee, published in the New England Journal 2013, that leads to very clear molecular markers, to a landscape of AML with respect to the cytogenetic changes as well as the molecular genetic changes. And by using this, now we also have specific drugs that can be applied and address such pathways, such genes, and are more and more of interest in the future. So one prime example that we know now since nearly 20 years is this specific subtype of acute promyelocytic leukemia, and we can target the PML-RARA gene fusion by ultrans retinoic acid, or today even arsenic, and after 3 weeks of taking these drugs, the bone marrow looks like that because the cells start to mature. So that I want to show just with this one example how important it is to describe and to discriminate the molecular structures in leukemias and lymphomas by using techniques that are now more and more next-generation sequencing. And if we go back to this slide, you here see how many different drugs or antibodies now can target specific molecular or cytogenetic changes within AML patients. And we have to be aware that, for example, mutations in the spliceosome have been found to be of importance 5 years ago, and now, after 5 years, there are already drugs available that can be applied in patients' phase 3 trials. So this again demonstrates how important it is to investigate those genes, those targets for more and more specific targeted treatment. And I will come back to this later. And the initiative, for example, in the US called Precision Medicine or the Moonshot, and how this is now going to be implemented in a routine setting. So just to give you an idea what happened in the last 10 years in our lab and what we experience in this time. So I just have 3 different techniques here: the flow, the fish on interface or metaphase fish, and the genes that we had in our portfolio. So with respect to the flow, we started with 5 colors. Now we use in a routine setting 10 colors. We started with 54 different markers. Now we have 60 markers. So there is no big increase of new information. It is still of interest and very important to use this, but this is what happened. With respect to the fish probes, we used 50 fish probes of different targets or different mutations. 10 years ago, now we have 131. But what happened to the genes we need to investigate by any of the molecular methods? So 10 years ago, we offered 37 genes of interest that should be investigated in a routine setting at diagnosis or for minimal residual disease as well. And today the number is 234. And they can be investigated in around 600 different assays, because for one gene, we sometimes have 3 or even 4 different assays, depending on the respective question, or being investigated at diagnosis or for minimal residual disease, we need different approaches to investigate the same gene. So you, I think, clearly can see where the increase of workload as well as information has come up, and that makes it more and more important to have new techniques to go forward. I also would like to mention just on this slide here that we accredit also next-generation sequencing in our laboratory 6 years ago now. So all the investigations we do, if we not start with a new assay, but after that, when the assay has been tested in the laboratory according to the ISO norms and the guidelines in the ISO norms, then all the assays are accredited. That means that all the assays we do by using next-generation sequencing follow very strict guidelines, and the results and the assays are accredited according to ISO 15189, what is the highest accreditation in Europe. So I just told you before, and this is just a short summary at that time, that these molecular markers matter because they lead to really actionable results for the patient's treatment. Not only at diagnosis, but for classification, for prognostication, and for targeted treatment, and for follow-up during the course of the disease, after transplantation, and with many new drugs today, we also can reach very low levels or even zero of their respective molecular marker that we found and picked up at diagnosis. And that even leads in the CML setting to stop the treatment. And if you do this, you even more need to use molecular techniques to be sure that you would pick up the relapse, the molecular relapse of the patient on a very early time point to then start with the treatment again. So with respect to the instruments and the assays we use in cooperation with the next-generation sequencing workflow, this slide shows you the access array chip with its controller, and you can pipe it 48 samples into the left column and up to 10 multiplexed primers in each of the 48 primer wells on the right column here. Thus, it leads to up to 480 amplicons prepared for each sample, and this access array provides a simple and fast approach to prepare a large number of amplicons for 48 patient samples. And additionally, it provides a high degree of flexibility because as the primer plates could be prepared individually for each access array, you can just change your mind with the next assay or the next experiment. Thus, each array could contain a unique panel with a distinct set of amplicons or genes you want to investigate. Another technique that we used many years is from RainDrop technologies, and this uses the genomic DNA from the patients and the primer pairs in up to 3 million little droplets. And due to electroevaporation, you can now put the DNA into the droplets and then go for PCR and sequencing. So this is one of the very important instruments we also use in our next generation sequencing setting. And another approach is not only to use the instrument for droplet generation and PCR, but also for sequencing, or what is more and more of interest, as I told you before, for minimal residual disease, because if you pick up a specific mutation at time point of diagnosis, you can follow up the patient, and the final readout looks a little bit like the readout from the flow instrument, and the detection level is around 1 to 10,000 or 1 to 100,000. So the sensitivity is in the minimal residual disease level, and I think this will be one of the most important advantages in the future. The next slide shows you some of our experiments we conducted in a cohort of 18 patients with a known NPM1 aberration. And we compared the performance of the RainDrop systems to the Fluidigm EP1 instrument for the digital PCR applications. So the results from the RainDrop instrument are depicted in red, whereas the gray outlines the results reported from the EP1 system. The higher the aberration intensity, the more concordant the data is. You can see that detection thresholds for the EP1 system is reached at around 0.1%, whereas the RainDrop system also detects aberration one magnitude below at 0.01%. And another experiment we used to compare and generate data through sequencing on a 454 instrument with results generated on the RainDrop digital PCR application in one run and a subsequent replicate. So concordance between both platforms is extremely high within all three sampling points. The last three samples were patients in progression. That means that we had data from another previous time point and knew that the patient was bearing this specific mutation. These three samples turned out to be negative in our initial 4x4 analysis, but at a very low level, mutation was picked up through the digital PCR, demonstrating the increase of sensitivity by nearly 1 magnitude as well. And here you see that we did some dilution series observed here with the RainDrop load against the regression line, and another dilution series here with the experiment, the observed and the expected dilution sensitivity. And I think it is clearly shown that these instruments work for the measurement and the detection of minimal residual disease. So during the last years, we moved forward with at least 3 different steps. So we combined the instruments I showed you before and the assays, and the turnaround time was around 6 to 7 working days. The input of DNA was 2.2 microgram at the beginning, we in parallel analyzed 489 amplicons, 10 samples per run for covering 31 genes of interest with sometimes the complete coding regions or hotspots of the respective genes. So the next step we did was we decreased the turnaround time to 4 working days. The input of DNA was only less than 100 nanogram. The number of amplicons as well as the samples and the genes increased for this next platform, combining the RainDance Thunderbolts and the MySeq from Illumina. And today we again moved to the next platform using these fluid-line access areas I explained to you before. The turnaround time is around 4 to 5 days. The input is very low, only 18 nanogram of DNA. And we analyze up to 75 patients per run and 47 patients in an array that is processed, as I told you before. So this is what we do, and this is an idea how the turnaround time is built up. Just starting, for example, on a Monday, receiving the sample in the lab and starting with the FICOL or the lysis, then followed by the DNA isolation, fragmentation, and then the thunderstorm or the fluid-line access array, breaking, purification, quantification, and then the runtime of the instrument, of the MySeq, then the end of the sequencing, and then what is a very important aspect I will show to you a little bit later, the data analysis and writing the clinical report. So around 1 week, it will be quicker, hopefully very soon, but it will not be within hours because the different steps and procedures need to be done. The timeline of establishing NGS in our lab is depicted here. We initially started with the 4x4 flex and unit systems and screening for single distinct genes in our patients. We quickly felt the need to expand to panels as the majority of the entities in leukemias are characterized in a variety of genes. Screening for them one by one is time-consuming and expensive. So we decided to integrate the then upcoming MySeq in 2011 with a significantly larger throughput and lower cost per base when the SPS chemistry was applicable. We implemented routine diagnostics with a Myeloid, including 26 genes, and a lymphatic, including 13 genes panel, using the RainDance technology based on library preparation method, using the thunderstorm instrument. It employs a microdroplet technique to forcibly create up to 1,400 amplicons per sample in an overnight process. To improve upon throughput and also decrease the turnaround time, we now move to the next instrument at the end of 2015, exploring exome and whole transcriptome sequencing. To reduce the need for such genetic analysis, we also are looking into gene fusion detection with the RNA access library preparation protocol. So on the next slide, you see that at the end of last year, we conducted a beta site test for the TruSeq RNA pad capture panel. It targets 1,385 genes associated with a variety of capsules and thus enables the comprehensive analysis of the cancer transcriptome. It also includes a number of known fusion transcripts. It uses surprisingly low input material and enriches all transcripts using a probe-basis capture method. Briefly, biotinylated probes hybridize to the target regions of interest and are then captured within stressed RPD beads and subsequently enriched prior to sequencing. Library can be used for gene expression analysis as well, variant calling, and known as well as the de novo fusion detection. And the first results are very promising in our hands. So the next slide shows that during our beta-site evaluation, we conducted a series of 3 runs with 12 samples with known rearrangements within orthogonal method. For all samples, we had cytogenetics and FISH data available already from the standard routine, as well as real-time PCR results, which were acquired also during our routine diagnostic approach. This slide depicts the 6 samples of our first run. On the left, you see the known rearrangements, and on the right, all fusion transcripts, which were screened for these respective samples. So the next slide then shows how we use the RNA-seq alignment app base for alignment and variant calling, and we're very positively surprised that all known rearrangements from our orthogonal approach were picked up by this sequencing approach. We performed alignment of the sequencing data with STAR and used MANTA for fusion detection. Data across all samples were very homogeneous as depicted in the screenshots. Run was performed as a 2 times 76 base per run. The same app was also used for variant calling. We had some data for the same set of samples with previous Amplicon sequencing of DNA and were able to detect the majority of the mutation through RNA-seq as well. So here are some results of the gene expression analysis from the same run. Again, we use the base app for computation, RNA Express. Each run consisted of 2 controlled samples in addition to 6 patients, so we were able to perform expression analysis immediate to completion of the run without additional information. The app computes significantly mutated genes between samples and controls and potential correlations between samples in a heat map plot. Currently in design phase is a Myeloid panel with a TruSeq custom Amplicon, a low input protocol. It will consist of 27 genes covering 118 exons, which are known to be mutated in myeloid malignancies. It is dual-stranded in contrast to the more traditional paired end sequencing, where we were able to truly distinguish between the forward and the reverse strand, as they are all separately sequenced. We hope to be able to filter out sequencing artifacts more easily and also lower the variant calling threshold from around 3 to a region close to 1%. Some genes are covered for the complete coding regions as they are known to be mutated in lots of loci throughout the gene, whereas some other genes have specific hotspots associated with a certain entity. So this is all only possible using a very comprehensive and elaborated data analysis pipeline that we built up together with our bioinformaticians over the last 6 years. And this pipeline not only follows the sample from the start, coming into the limbs and all the other steps and procedures before the next generation sequencing instrument gets into place, but also very importantly helps us to analyze the outcome of the data after the next generation sequencing by the implementation of several bioinformatic tools as shown here for the functional interpretation, especially of the variants. And without that, it is very difficult to use data from next generation sequencing. As far as we think here, and this is my opinion as well, because you need to be aware that you will pick up a lot of variants of unknown significance when you start, and especially when you don't have experience with your own sample quality and your own sample entities. And during the process of analyzing your leukemias, lymphomas, or whatever, you will finally get to more information in-house and need to compare this information that you collect over the time with databases that can be used for this. So how does it look like finally if we pick up a mutation in P53? It is by the help of our bioinformatics pipeline compared with the COSMIC database. We use polythane and SIFT to look about the consequences and the prediction of the mutation. We see the same mutation in the IRC database as well as Clin Var and dbSNP as well. So I think without this help of the available databases, it will be difficult, at least in a routine setting that we are, and for accredited routine laboratories to let those data get any influence into written reports that go for patients. And just to make sure and give you an idea how difficult it is to depend on the curated databases that are now web-based and are available, this is one example from the Clin Var. And if you see a mutation like that in Clin Var, you have to be sure and you have to be aware that only 12% of the interpreted variants in Clin Var have 2 or more submitters right now. And even if you have 2 or more submitters, in 21% of those, you will have different interpretation of the variant that was picked up. Means that some interpreter will say, well, this is a damaging mutation, and others will say, well, this is a benign change. So even if we believe in these data sets that are web-based and that can be used for the interpretation of your data, we have to be sure that this is at the beginning to be used for a routine report setting. So in instances where we do not have any or insufficient information in databases, we are forced to rely on computational methods to determine the impact of a certain variant on a protein function. So I won't go into details too much here. The purpose of this slide is only to show the availability of a variety of tools to be incorporated into a variant classification pipeline. The most popular among this list are, as I told you before, polythene 2 and SIFT tool, predicting possible impact of amino acid substitutions on the structure and function of a human protein using straightforward physical and evolutionary comparative considerations. So, just as an example, several tools were developed in the recent years, each with its distinct approach. As they each have their strengths and weaknesses with certain biases, generally published comparisons have similar findings. However, there are still many variants with uncertain or contradicting consequences, as I lined out before, together with the fact that no database entries exist. That can happen as well. Interpretation turns out to be very hard. Most algorithms will predict about 10 to 20% of SNPs to be damaging, for example. So the most recent recommendations for variant classifications from the American College of Medical Genetics recommends always using multiple prediction tools and deriving a consensus result to be written in the final report for the patient. It is also important to select tools producing results from different approaches. That is, for example, protein sequence homologue and evolutionary conservation. And the different approaches taken here as shown on the next slide by the EXAC consortium. They assembled a collection of around 60,000 exome sequencing and data, which has been processed by the same pipeline. They distinguish between good and poorly covered regions and then derive population frequency data for each position in the exome. This approach is in between the database and the computational approach in regard to being able to classify variants at an arbitrary position. This plot depicts the coverage for each exon. For example, in the RUNX1 gene, you can see that the exon 1 is not covered well with the commercially available exome sequencing kits due to its high GC content. Below the coverage plot, you can see all variants in RUNX1 picked up by the pipeline with their respective allele count and frequency. This is a convenient method to assess the degree of pathogenicity of variants not described so far in any public database. So coming back to our experience over the last 5 years now, as I told you before, we started with the 454 Roche instruments and then moved over to Illumina in the last years. So all over in the last 5 years, we investigated more than 180,000 genes by one of these two techniques for next-generation sequencing, and 2/3 of them have been analyzed by the Illumina technique. And if you ask yourself the question, what genes were in the top 30, this is the top 30 out of 120 different genes that we analyzed. And the most often gene was the RUNX1 gene that we investigated by next-generation sequencing tools more than 15,000 times for research as well as for routine approaches in the last 5 years only. So how should a molecular workflow maybe look like today and in the next years? So starting with the malignant cells and preparing DNA as well as RNA-specific pipelines and assays, we can add to the respective DNA or RNA of the patient sample-specific indexes and then go for panel or exome sequencing or even RNA-seq to pick up not only the mutations or the copy number variations, but also in the same run, in the same approach, analyze for fusion genes, expression analysis, and mutations on the cDNA level. And then, by the help of several parallel software tools, may finally come to reports that include the results and even a recommendation maybe for treatment after. And before I go further, I think, Svetlana, you would like to ask this poll question to the audience.
Yes, thank you, Professor Haferlach. We would like to ask our audience this one quick question. Where do you believe is the biggest challenge for adopting NGS into your laboratory? Please select one of the answers below. A, initial investment for instrument purchase. B, lack of NGS experience. C, lack of bioinformatics support. D, availability of optimized gene panels. Or E, time required for assay validation. Professor Haferlach, please feel free to continue with the next slide, and then I will come back with the results.
Okay, thank you, Svetlana. So the next slide, again, demonstrates what I explained to you before, that from our perspective, in the very next future, the next-generation sequencing platforms and instruments will help us to get all this information that we now get out of 3, 4, 5 different techniques, instruments, and in parallel workflows only in one approach by using these sample-specific indices. And maybe, Svetlana, you have already any information what the colleagues in the webinar think would be the most difficult aspects?
Yes, so we just got the results. So we have 55% of our attendees thinking that the lack of bioinformatics support is one of the biggest challenges. And the second one would be the time required for assay validation. So I don't know if you would like to make maybe a comment or two about these results of this question.
Yeah, well, thank you. Thank you all that took time to answer this very short question. I really think that this is true because it is easier today to get an instrument installed in your laboratory and maybe buy an assay that is already available in the market than to make the data really valid for especially a routine approach. So this problem of the technique and the instrument, that is more or less nearly solved, but it is very hard to do the data interpretation as far as we experienced it over the last years. And that will be not solved in the next 1 or 2 years, but we all have to be very cautious and careful to not overinterpret what we see by applying such platforms. And for example, it may be easy to do exome sequencing today, but what are you going to do with the data when you really want it not only for a research question, but if you want it for a routine patient, and finally on that report, the treatment will be decided accordingly. This is really, from my perspective, an aspect that needs much more time and efforts, and I can fully agree with you that the bioinformatic pipeline is behind the technical pipeline of these techniques. So we all have to work on that, and I'll also try to do this within the European Leukemia Network as well as within the Task Force for Precision Medicine at the ASH, where we also try to get input and give our input how to go forward with the bioinformatic approach. I would like to share with you a very short aspect that is also upcoming for the future, how to store. Because we don't want to throw the samples away, because we want to backtrack, we want to look in the samples 4 or 5 years ago, we want to do research. And so we started to introduce a new instrument a year ago now in our laboratory that changed the way how to store DNA or RNA pellets or cell pellets on -80 degrees centigrade. It looks like that before, and it looks like that since the last 9 years with all these freezers where we store the samples. But starting with these freezers like this, the instrument now we use looks like that, and this is using a 2D barcoded system that automatically cap or decap the respective vials, is readable due to these 2D barcodes here by specific readers, and then the vials and the samples go automatically into this big room with -80 degrees centigrade, and so we are able to store our cell pellets, DNA as well as RNA, due to this instrument that is really helpful for large-scale approaches in a routine in the future to store your DNA, RNA, and then go forward. So why is all this needed? I told you before, we have more and more knowledge about targets. We will focus something that is called personalized medicine, or choosing wisely, as the doctors say right now, or precision medicine. What is this precision medicine? It is a new wording that was brought up by the President of the United States in January last year in 2015. And what is going on there, and the discussion is worldwide from this initiative from Obama, is to enable a new era of medicine through research and technology and the policies. That is also very important to empower patients, researchers, and providers like Illumina or others to work together towards a development of an individual treatment. And what the US is doing is what we also are doing right now, because we need money for the instruments, we need money for research institutes, we need money for the rules, how to work on that, the regulatory structure, how to do interlaboratory trials, how to test, how to validate an assay that not tests only one gene, but 200 genes, 100 genes, even the exome. And we need support of the software. And we have to say that this is only a less amount of money that goes here, and we all think that there must be more. There is a more general debate also on genomics in Europe and worldwide, and the question is, where should it be placed? Where should these instruments, where should all the infrastructure be built up? At the hospital where the patient is, or only in academic centers with all the background of the scientific community, only preferably in study groups or centralized per country or even Europe-wide. So there's a lot of discussion needed for the future that came up by the availability of next-generation sequencing techniques. And this is not all, but there's also patent issues to be discussed and solved, because if we are going to investigate 100 or 200 genes in one run for one patient, and some of the genes have patents, how should we deal with this? What is the way to work with the patent owners? This is still an unsolved aspect. I address also accreditation and ring trials. There is reimbursement, a real problem we also had in Germany over the last 3 years, how to get this reimbursed. There is an open question still not solved. And maybe also the discussion is that if there is a companion diagnostic that is needed to be applied before a specific drug can be given to the patient, should the company that owns the drug also pay the companion diagnostics? So there are a lot of open questions right now. And coming back and going to conclude, currently we are equipped with 3 MiSeqs and 1 NextSeq instruments, and we initially started with the MiSeqs to our routine screening with a screening of the entity-specific panels, as I told you before, like a Myeloid panel or a lymphatic panel, and now included the NextSeq in our lineup to decrease the processing time as well as to multiplex samples for exome sequencing and transcriptome analysis as well. It also gives us now the flexibility to increase the number of Amplicons per panel without sacrificing samples throughput. It would be nice to see an increase in the read length, I have to admit, on the NextSeq to match up with the MiSeq to be able to sequence the same set of Amplicons on both instruments as well. I would like to share also with you this information just to show you where these visionaries think about right now. They started to build up and to found a new company called Grail. It's a startup with Jay Flatley, the CEO from Illumina, and Bill Gates, and Jeff Bezos from Amazon. And they started this 2 months ago, and they are going to build up a single blood test for all cancers, and they think that they can go into the market in 3 years from now. So we are today, even if we think we have a lot of specific techniques, we may be beginners in 3 to 5 years from now by such new options that are available right now. So this is the microscope of Rudolf Virchow, a very famous pathologist in the 19th century in Germany. And this is nearly 200 years ago, and what happened in the last 50 years is this, that using morphology, cytogenetics, FISH, immunophenotyping, and standard molecular assays, we now are in an era where we can, in a routine setting, apply all these techniques for exomes or even genomes. And I think it's a fascinating time to do this, and I'm looking forward to use this more and more in a routine setting. The turnaround time is decreasing, as you see here, with up to 5 working days with next-generation sequencing. This is good for many aspects in a routine setting. So in conclusion, this is my last slide, I think that the use of next-generation sequencing is flexible to address hematological cancers according to their respective entity. The turnaround time is getting more and more acceptable and will decrease over the next 1 or 2 years as well. The workflow can be implemented in a routine lab and can be accredited as well. And the comprehensive approach leading to precision medicine, this is the basis, the next-generation sequencing approach. However, there are a lot of open questions: which platform or assay to use, DNA and RNA-based assays in parallel. The sensitivity has to be better for really minimal residual disease if you want to rely only on next-generation sequencing platform. The reimbursement and a lot of ethical questions that I did not go into detail in this talk also have to be discussed within the community. So I would like to thank you all for joining, and I would hand back to Svetlana for questions and answers.
Thank you very much, Professor Haferlach, for this very interesting presentation. First question, Professor Haferlach, could be, what is the most valuable benefit that NGS has brought your laboratory in terms of precision, sensitivity, efficiency, and or time saving?
Well, I think the sensitivity is going down to the minimal residual disease detection already, and this is of importance for a lot of molecular assays that we need to run for our patients. And what is also very important is that we significantly have a higher throughput of the samples by using such platforms. We also see that it's easier with respect to a variant calling for complex mutations, especially for frameshift mutations. And what I also see as another advantage is that the allele information is now derivable, which may finally not only broaden our understanding of the respective pathobiology of the disease, but also may influence our therapy very soon. And so there are a lot of advantages using next-generation sequencing, even if you just think about a routine application.
Great. Thank you. So another interesting question is in regards to validation. So do you continue to validate pathogenic variants using Sanger sequencing?
Well, this we did a long time, and we are also asked how about this procedure, and the answer is no. Indeed, it is the contrary, because we now reach a point where, at least in our hands and due to the many years and many samples, the validation is redundant or even impossible, because Sanger has a mutational calling thresholds of around 10% maybe, and we frequently observe mutations below that threshold. And so especially in patients with progression or question of relapse, where we have prior knowledge about the loci of the mutation, we need next-generation sequencing and not Sanger sequencing to pick it up very early, and our current threshold in these patients is around 1%, which cannot be reproduced by a Sanger sequencing. So as outlined in my talk before, we are now forced to perform Sanger sequencing when we want to get it reimbursed, and I hope that will change in the next future. But with respect to all the other questions, we go for next-generation sequencing and do not validate the findings with Sanger sequencing anymore.
Okay, great. So we just got a couple of additional questions. I think we will take one more question, and the remaining questions we will try to send you by email. So the last question could be, what should be the minimal base coverage for mutation detection in de novo AML samples for minor clone detection?
Well, we are going to do this with 200 reads at least, but this is also a question of the respective lab flow, and the median coverage we normally reach here is 1,500 in our routine approach, so we are quite higher with this. So in contrast to genome sequencing or exome sequencing, we normally reach a coverage of 1,500, but I would suggest to be at least with 200 reads to address this question that was just pointed out.
Okay, that's brilliant. Well, thank you very much, Professor Haferlach, and thank you to our attendees for sending us through some very interesting questions. And with this, I think we can close today's webinar. Thank you, Professor Haferlach, for your excellent talk, and thank you to all of our attendees. Goodbye.